Good afternoon, everybody. This is Daryl Diamond from the Association of Community Cancer Centers. I want to thank you all for joining us today and welcome you to today's webinar, which is the future of molecular testing, optimizing efficiency under increasing expectations. This is part of our molecular testing peer-to-peer webinar series. Funding and support for this series is provided by Pfizer Oncology. This webinar will be presented by Dr. Joe Kim and will feature Dr. Randall Oyer. Dr. Oyer is the Oncology Medical Director at Lancaster General Hospital in Pennsylvania and has served on the Advisory Committee for ACCC's Molecular Testing Education Initiatives. Dr. Kim is the President of Medical Communications Media. Just a couple of housekeeping reminders for today's webinar. If you look at your screen, you're going to notice a control panel on the right-hand side, and you'll see a chat box or a questions box. We are going to be taking questions live during the broadcast. So feel free to type them in there and we will get to as many as possible during today's webinar. All the questions that we don't get to also we are going to collect and we will send them to Dr. Kim and Dr. Oyer to answer for you after the webinar. Also note that it's going to be an email sent to all registrants within the next few business days advising where to access the archive for this webinar as well as all the other webinars in the molecular testing series. At this time, I want to introduce Dr. Joe Kim.
Great, thanks very much. And I want to thank everyone for joining us on this webinar. We're excited to present information and give an update on some ongoing projects as well as a recent project that we completed with ACCC. But before we get started, let me ask Dr. Oyer to introduce yourself as well as just tell us a little bit more about Lancaster General and your cancer program there.
Well, good afternoon. Thanks, Joe, and thanks, Daryl, for the invitation to speak with you this afternoon and to participate with the ACCC in this important webinar. I am a medical oncologist, and I work at Lancaster General in Lancaster, Pennsylvania. At the core of our system is a 150-year-old, 630-bed hospital. We're an American College of Surgeons COC-accredited program. And in 2013, we added a freestanding cancer institute, the Ann B. Barshinger Cancer Institute, where we see approximately 2,000 new cancer patients annually. And as of this week, we become a part of Penn Medicine of the University of Pennsylvania in Philadelphia, the fifth hospital in the University of Pennsylvania system.
Great. Well, sounds like there's definitely a lot of interesting and exciting changes ahead. So. Well, we'll dive right in, and I'll start by introducing the project that we were involved in, which was the 2014 Molecular Testing in the Community Oncology Setting. This project involved us going out to 8 different cancer programs around the country, and we performed these process improvement learning labs. Each hospital had their biomarker testing processes and baseline results analyzed, and we provided that to an advisory committee, in which case Dr. Oyer served on that advisory committee. And then we actually went in and spent 2 hours with the entire multidisciplinary team talking about their process, how they communicate, how things work, in terms of the clinical workflow and how things are done and who orders a test. And we spent a lot of time analyzing and reviewing their own data as well as coming up with suggestions for improvement. Every center then used the Plan, Do, Study, Act, or PDSA cycle for improvement as the framework to identify and implement some process changes, and then we followed up with every center in 3 months to see what they had achieved as well as what they had planned moving on in the future. This slide here lists those 8 centers, and you'll see that they are all around the country. And there were other programs similar to this where we went out to other centers as well, so we've collected now over the years a wealth of information, knowledge, as well as practice patterns and trends in terms of what's actually happening in the community as it relates to biomarker testing in advanced lung cancer. If we de-identify these 8 centers, and if we cluster them into those who are treating more than 100 lung cancer patients a year versus those that are treating fewer than 100 lung cancer patients a year, you'll see that for EGFR and ALK testing rates specific to patients with stage 4 adenocarcinoma of the lung, that the rates vary from the 40 to 50% all the way up to the 60% range. Now keep in mind that this baseline information was predominantly from 2013. Some of it was from the late part of 2012, but it really does show that even at every center in the community, there's room for improvement. And during the learning labs, we presented each center with their own data so that they could actually see what their testing rate was. They could see their lung cancer volume and then have that discussion with the entire multidisciplinary team. So today, we're actually going to be talking about some of the lessons learned in terms of some key barriers that we identified, as well as improvement plans that some of these hospitals incorporated. And we know that we're living in an era where biomarker-driven medicine is becoming more and more predominant. There are growing opportunities to collaborate in various ways with research centers to leverage their expertise. And we also know that lung cancer screening is going to bring even more lung cancer patients into the volume of these busy hospitals. So, you know, how are pathology departments dealing with things related to biomarker testing? And, you know, how are these guidelines evolving? We'll be touching upon some of those things as well. Now, in 2013, the College of American Pathologists, or CAP, the International Association for the Study of Lung Cancer, IASLC, and the Association for Molecular Pathology, AMP, released guidelines on the selection of EGFR and ALK tyrosine kinase inhibitors for these lung cancer patients. This guideline really formed the basis for a lot of the discussion during the learning labs, because the learning labs were an interdisciplinary team. These guidelines are primarily being driven by the pathology community. And so we spent a lot of time, and we'll touch on some of the key points. But one thing that I do want to note is that earlier this year, these three organizations released a press release announcing that they recognize that much has changed in this field. And so they're planning on updating these guidelines, and they anticipate that those guidelines updated will come out in 2016, addressing key issues such as ALK testing recommendations, EGFR and ALK resistance in these lung cancers, as well as emerging molecular targets and the growing need to rebiopsy when the disease actually relapses. So we know that non-small cell lung cancer is a very heterogeneous disease. And historically, back in 1987, we only were aware, the medical community was only aware of the KRAS mutation as a driver mutation that led to this cancer. We knew that there were probably a host of others, we just hadn't identified them yet. Then in 2004, we discovered EGFR, and come 2013, now you'll see that there's a whole list of these driver mutations that we know contributes to the pathogenesis of lung cancer, and there are a number of different targeted therapies aiming to see what kind of impact and effect it has on these various targets. So going back to the guidelines from 2013 that CAP, IASLC, and AMP released, a key question that we used as a basis of discussion during these learning labs was, when should a patient specimen be tested for EGFR or ALK mutations? And the guidelines clearly indicate that any lung cancer patient that has an adenocarcinoma component who has stage 4 disease should be tested, because if the mutation is positive, there are available therapies that are FDA approved that have shown clinical benefit. They also recommend that the tissue that's obtained during biopsy needs to be prioritized for EGFR and ALK testing. So we had a lot of discussions around what kinds of processes are in place to ensure that adequate tissue is being obtained, as well as that the tissue is being prioritized, and that the appropriate tests are actually being ordered in a timely fashion without significant delay. And that was really, there were a lot of different discussions and ideas that came from that. We also know that the NCCN guidelines, which continue to get updated, also support the EGFR and ALK testing in adenocarcinoma patients. We know that NCCN strongly advocates for broader testing and enrolling patients into clinical trials and looking for those opportunities as well. And that became a critical component of our discussion at cancer programs where there was an active component of trying to identify patients for clinical studies. So when it comes to some of the key barriers that these different programs in the community are dealing with, we know that there's a variety of them. We've now had actually several webinars on this topic addressing some of the other barriers related, for instance, to the tissue acquisition, as well as processes and teamwork and communication. Today, we're really going to be focusing on some of the other process-related barriers, such as, should the molecular testing get delayed until you know the disease staging? When the initial biopsy is performed and you establish that this patient now has lung cancer, A lot of times, the patient hasn't been staged yet. So should you wait for that staging information, or should you just go ahead and test that specimen for EGFR and ALK mutations? So that became a question as well as a key discussion point. Another one was around the types of tests that should be ordered. Should it just be limited to EGFR and ALK? Should it be broader than that? And how is every cancer program in the community dealing with these questions when some of the medical oncologists potentially want additional tests beyond EGFR and ALK? How can you standardize the process? Another issue that came up was regarding who should be the ordering physician when it comes to ordering these biomarkers. Should it be the pathologist or the medical oncologist? Because often when the pathologist is reviewing the biopsy, the patient hasn't even seen a medical oncologist yet and hasn't been assigned to one. So, what is the right process to expedite that testing and to delay and to remove the delays? And then finally, there was the issue of the Medicare 14-day rule for inpatients who actually get the first diagnosis of lung cancer as a hospitalized patient. And within the Medicare framework, if you then perform biomarker testing while the patient is still in the hospital, you risk not getting reimbursed. You have to wait till the patient gets discharged, and then 14 days later, you can then send that specimen off for the biomarker testing in order to get reimbursed by Medicare. So once again, do you wait the 14 days and possibly longer because the patient might be in the hospital for an extended period of time? So these were some of the key barriers. And I guess, Dr. Oyer, I would love to hear your both perspective as well as experience in terms of dealing with these from your role in the advisory committee as well as your role within the Lancaster General Hospital and Health System and the cancer program there.
Thanks, Joe. I certainly agree and recognize all the steps that you went through in your slides. I mean, you sort of started in 2014, 2013, and touched on this step, but I feel like we've been on the same road of discovery together with the ACCC since 2010, 2011. In 2010, we looked here at our cancer program at molecular testing and felt that we were dealing sort of with the Wild West, that there were patients with metastatic non-small cell lung cancer who weren't getting the testing they needed. We had patients getting broad testing that didn't have clinical applicability. And we recognize the importance of providing a unified and up-to-date approach across the health system. So we developed a diagnostics committee, which was co-chaired by the chairpersons of diagnostic radiology and pathology. And this was a committee that was charged with developing a framework for selecting diagnostic testing based upon clinical evidence and for developing the processes to make sure that the testing was available across our system in a timely and consistent way. We needed a centralized body to develop safe and effective processes for obtaining the tissue and tissue prioritization, as you mentioned, in using certain quality standards and understanding what the reimbursement issues were. And at the very first meeting in early 2011, our diagnostics committee selected molecular and genetic testing as the first area that it was going to study across our system, and they requested a clinical presentation on testing in lung cancer from our lung cancer steering committee. And as it turned out, at the same time, the ACCC developed a new initiative to do an inventory of what was known at the time about molecular testing in non-small cell lung cancer, and actually sent out a letter seeking participation. And we thought about that, and we decided that based upon our own goals and the workload we had, that the goals of the ACCC program were really the same as our own, how to improve patient outcome and improve the value of what we do. And that since the ACCC had really been a trusted partner in other programs, that we could probably do this better with the input of the ACCC and the other programs that participated rather than do it on our own. And that turned out to be the case.
So panel where there's consensus from your team of oncologists, these are the biomarkers that we want to test for, and here's why. And can you just tell the audience about how you came about that panel approach for biomarker testing in lung cancer?
Sure. I'll tell you what the final state is, and I believe it's going to continually be in evolution. Before I tell you about what's on the panel, I think one of the important lessons that we learned was that what was on the panel, or what we were testing for, would be fluid as new information became available. But we wanted to develop a really solid process where we had clinical involvement in terms of guidelines, quality, metrics, etc., but that we needed to have panels or tests that were continually updated. And so we went from doing just EGFR with reflex to ALK to developing a 6-test panel for patients who had stage 4 non-small cell lung cancer, particularly adenocarcinoma. And we're doing next generation sequencing for EGFR, KRAS, and BRAF, and FISH testing for ALK, RET, and ROS1. We do this through our partner at the University of Pennsylvania. It's been really the perfect marriage of a clinical program and an academic medical center, where we understood at the academic medical center that they had a solid tumor panel that had 44 genes. Our clinicians were concerned about the cost. Our clinicians were concerned about explaining to patients what all this meant and having a lot of information that was not actionable. And our doctors, in close consultation with our colleagues at Penn, felt that these were the 6 tests that were clinically actionable. We're now rethinking this, and I mentioned being fluid, the possibility of adding MET and HER2, because there are FDA-approved drugs that have these as targets as well. So I think it's going to be a fluid process. And
yeah, it's
so
an
our
interesting
process
point, right? Because as you mentioned, it's always changing, and it seems like whether you're meeting quarterly to establish these or whether it's monthly, there's really not an ideal time frame, because the data continues to evolve and change. But we're certainly looking forward to hearing more about that in upcoming webinars and presentations. Now, how about the Medicare 14-day rule for inpatients? I know that we spent a little bit of time discussing this, and within your organization, how have you dealt with this issue of, should you wait before the patient gets discharged and wait the 14 days, or do you go ahead and just send out for the biomarker testing if the patient is diagnosed as an inpatient?
We've spent a lot of time talking about this, and our leadership has basically said to us, if you need this test to take care of somebody who's in the hospital or soon after they're hospitalized, go ahead and order it. We cannot put the cost or the financial implications first. We do not have a fast rule that says don't do this for 14 days. You may get a call from the lab that says, do you need this within the next 14 days? If so, we'll proceed. If not, would you mind ordering this as an outpatient? Many times, the patient needs additional stabilization or needs to complete their diagnostic workup, and we'll take that time and do it as an outpatient. But it's not a hard stop for us. We're aware of it, but we just really proceed putting patient care first.
Sure. And it seems like certainly in the community there are a number of organizations that are probably going to see an increase of the outpatient diagnosis as opposed to these patients being diagnosed as inpatients, especially with lung cancer screening programs in place. So let me move on to this next slide, which talks about some of the issues around the tissue handling and the biopsies, as well as just seeing if the tissue is adequate. Because on this slide we also mentioned some things related to policies and procedures. But before I get into that, tell us a little bit about the discussions that your pathologists, radiologists, and surgeons have had around lung biopsying and making sure that the specimens are adequate for the pathologists, as well as making sure that there's enough tissue there for biomarker testing.
Well, we first took a look at who was doing the tissue procurement, and my recollection was that about half of the half was coming from radiology, and half and half from pulmonary and surgery. Although I don't remember those numbers off the top of my head, the key lesson that we learned was that there were different processes in place and different education needed to happen for different stakeholders. And we did a survey, for example, of our diagnostic radiologists, asking them which needles they were using, were they doing aspirates, biopsies, how many specimens were they obtaining, and we showed them on their own that they had a variability in their practice. They hadn't even really spoken to each other, and they learned lessons from each other, and we tracked back from the radiologist in his or her procedures what the success of the tissue specimen was. So just asking the question and making people aware that other people did it different ways, or that there was more than one way to do it, and letting people look at the data about what was most effective, and then raising the question about tissue adequacy led to, the Hawthorne effect. You look at it and there's already some improvement. Additionally, you mentioned earlier in your comments about tissue prioritization, and we just really made sure that everybody understood the concept of limited tissue capacity and prioritizing testing that was needed. And one of the lessons learned, I think, in the panel testing is that it's much more efficient. You get your tests and you don't have specimen exhaustion, which has been a real issue for us.
Sure, it's a great point. And I think going back to your comment about like the radiologists identifying variability even amongst themselves as a group, we found that trend as we went out to different hospitals and spoke with their teams. And there was even one hospital involved in the Learning Labs project where they had previously actually done an analysis amongst their radiology CT-guided lung biopsies, where they were switching from using fine needle aspiration, or FNA, to core needle biopsies. And there was concern from both the radiologists as well as other clinicians and administrators that switching to core needles might yield more complications. Well, they actually studied it within their own community hospital and found that within their own program and with their radiologists and with their volume of biopsies, that there was really no difference in complication rates between using fine needle aspirations versus these core needle biopsies. So I think every program took these issues to heart, and they really did want to raise that bar to make sure that now, instead of just obtaining enough tissue for the diagnosis of cancer, they needed to make sure that there was enough tissue there for biomarker testing as well. And we were able to share some of these stories with some of the other hospitals that were involved in this initiative. So I guess in terms of the idea of this, what is often called pathology-driven reflexive testing, it sounds like the panel approach that your organization has adopted is very much a reflexive panel. Are there cases where if someone says, you know what, beyond this panel, I would like to add on these additional mutation markers because I really think this patient might be qualified for this other clinical trial? Or are there times where Penn perhaps even makes those recommendations to your team?
We do additional testing for clinical trial eligibility as part of a clinical trial. Many of the molecularly driven trials have a tissue screening process, and the tissue goes to a central lab, and so that's become an automatic process that tissue that we expect and we look for tissue that may be part of clinical trial eligibility. If a physician requests molecular testing that is not part of our validated set, we will have a number of phone calls among people that we consider experts, our pathologists, our colleagues at Penn, the clinicians, and if we don't find if we're not able to figure out a clinically actionable step, we will provide the tissue block to the physician and let them take care of it on their own. That's our solution. If you're going to do something outside of what we think is clinically useful, we're certainly not going to stop you, but we're not going to do it in the lab. That's the point of having a diagnostics committee. That's the point of using evidence-based medicine. That's the point of having a consensus-driven process.
It's a great point, yeah, and I think that especially in the community setting, clearly these are the various sort of, human factor dynamics that we need to take into account because, we've seen hospitals where there may be 3 or 4 different oncology practices. There are still solo oncologists out there in the community as well as large groups. And when you've got this level of variation in terms of the kinds of clinicians who are practicing and treating patients, you're certainly going to get a wide range of opinions. So that's very understandable. Tell us more about the partnership between Penn. I know that you're now joining the Penn network, but prior to that, you had still had a established research partnership. And here under improvement plans, one of those is that these community hospitals are building or establishing either new or ongoing relationships with medical centers, either for the purposes of research or education or other things like that. Can you just share with us that journey and what it's looked like, as well as where you envision that that's going to go?
Lancaster General has been part of the Penn Cancer Network since 1994, was a founding member, and we've continually strengthened our relationship. Initially, it was a contractual situation where certain resources like access to clinical trials were provided, and we really strengthened the relationship when we built our Cancer Institute so that Penn was part of the quality, the planning process, the scope of services, to the point where we developed such a strong relationship around cancer that it really became a platform for other relationships across other service lines, and then really a relationship where we became integrated as a single institution. And I think that the broad strategy is that for people to be cared for properly in our country, people need access to care at home, a good, strong community hospital that provides longitudinal care, understands who they are as individuals, is there for emergency needs. But you need the support, the research support, standardization, sometimes quality, that you can get from an academic partner. And that's really been our goal, to make sure that we, as quickly as possible, transform new research findings into clinical care at home.
And I know that when I visited your new cancer center, you all have state-of-the-art video teleconferencing equipment. One of the models that currently exists out in the community are things like, for instance, these molecular tumor boards, where community hospitals are interfacing with these academic centers, talking with their genetics experts, their bioinformatics experts remotely, but looking at slides, looking at images together in sort of a collaborative fashion. Tell us how you're using your video conferencing equipment with Penn to have communication around either patient care or around education or research.
When we opened our Cancer Institute 2 years ago in July 2013, we installed video conferencing equipment that connected our downtown hospital and our suburban outpatient location, where we see about 1 million outpatient visits a year in the system, with the University of Pennsylvania, so that when we have a tumor board, we have 3 locations, 2 here in Lancaster and 1 at Penn, with clinicians in all 3 rooms, and not just at tumor board alone. But there are other people in those departments who are driving all that. But there's some areas where I think that teleconferencing makes a lot of sense.
Sure, and we're certainly seeing, I would say, overall a greater adoption of these kinds of technologies. Now, speaking of technology, I know that the theme of health information technology, or health IT, is something that seems to come up over and over. In many cases, people refer to health IT as an enabler. In other instances, it's really just become a barrier because of various challenges that hospitals and organizations and even departments have run into, whether it's an interoperability barrier or a health information exchange barrier. But there clearly are opportunities around things like big data and analytics, clinical decision support tools. How has the journey been at your organization in terms of, now that you're part of the Penn network, are you all making a lot of changes within your health IT infrastructure? Are you getting access to additional resources such as these either analytics or clinical decision support tools? Tell us a little bit about that.
Well, I think that we're past the point where health IT is a barrier. It is an enabler, and it has to be an enabler. And probably for most organizations and for older physicians like myself, it was a steep learning curve, a steep climb up to develop some level of proficiency. And I'm by no means suggesting that I am proficient, but at this point, it's no longer a barrier. It's an enabler. So in terms of working with Penn for several years, we've had care everywhere so that patients who were seen at Penn had access to medical records back and forth to physicians with a signature from the patient. At this point, being one integrated organization, we're making sure that we have the same versions of Epic. We're both on the same EMR, fortunately, that we have all of the 2014 versions of the clinical, the decision support, Beacon, the tumor staging, that we're really in sync. And our integration with Penn is finalized this Saturday, so on August 1st, we've just passed all of the regulatory steps. So this is really just the beginning. We've developed the clinical relationships, we've developed the programs where we complement each other, but the real IT integration, the health IT that's going to enable patient care, point of care decision support, and additional information gathering that's the foundation for research, that's only beginning.
Sure, and it sounds like it's going to be quite a journey. How about in terms of your the tumor registry? I know that within the Penn Medical Center, they have a tumor registry. You all have your own hospital registry. Are those going to get merged, or are you going to maintain different registries? Do you know what's going to happen there?
I think we're going to be working more closely together. We will be, I don't know what the final structure will be, but we will have one integrated system that looks at the same quality metrics, shares data back and forth, and develops a solution for incorporating molecular diagnostics into the tumor registry in a searchable field, just like T, N, and M, an updatable molecular diagnostics field, which becomes, the subject for query as well. Sure. Those are initiatives that we will do together.
Great. Well, we're wrapping up the webinar now, and we know that there's a lot of changes coming up in the future. I want to remind those on the line that if you all have questions, to please type them into your chat box so that we can address them. And we also know that, as we mentioned earlier, this, the evolving trend of this precision medicine, biomarker-driven medicine, and more patients enrolling in clinical trials. And I think even recently on the ACCC Network Forum, there's been some questions around, how do we get more patients involved in clinical trials? We know this is a COC accreditation requirement, and there's some, I think, creative ways and things that we've even learned within this initiative, and part of it being that just getting people to have that right mindset of identifying patients who might be eligible. And even right now within the whole oncology community, there's greater discussions around trying to get older patients enrolled. So I think that there are a lot of changing trends. We mentioned briefly about lung cancer screening, as well as these new and evolving biomarkers. And we also know that the whole discussion around value, whether you're referring to the ASCO value framework or other papers and publications and research looking at and measuring value in oncology care, we're going to see more and more about this. So I think that as we think about the future, clearly improving how patients with rare driver mutations are identified is going to be important, and this growing opportunity around clinical research, and the big data analytics, like the ASCO Cancer Link project and others. I think that there are some very interesting things coming along the way. So I guess I also want to remind everyone on the webinar that on the ACCC website, under Provider Resources, you will see a link for a resource called Ongoing Advances and Improvements in Molecular Testing. This is also going to get mailed out with the July-August Oncology Issues magazine. So you can both find it in print copy as well as online. But I do want to just address some of the questions that are now coming in with Dr. Oyer. And I guess one of the first questions here is around this whole precision medicine initiative announced by the President. How has this changed the tone of the conversations with your patients, or even changed how your providers and clinicians are having discussions with your patients, if at all? Can you tell us a little bit about that?
You bet. I think that precision medicine is an extremely important initiative that we will learn a lot. We will change our practices, and I think we're in the early stages of that. Honestly, I think that all advanced cancers should have molecular testing to the extent that it's known, and that the information that comes from that should be divided into 3 tiers. The first tier should be precision medicine or molecular diagnostics that are attached to FDA-approved therapies, and that should be clearly delineated. A second tier would be molecular diagnostics that could be used for clinical trials matching. And I think that that needs to be stored in some way, perhaps on the cloud, so that it can be updated as clinical trials become available, and you can have a match from that. And then thirdly, information that's just gathered and becomes longitudinal, observational to determine, that becomes a basis for research. Did the person turn out to have a good prognosis, respond to this drug, not respond to this drug? It sort of becomes the repository for study information going forward.
Sure. And another question that came in is just regarding lung cancer screening. Has your program had a screening program in place for a while, or is it new? And if so, how has it changed the volume of lung cancer patients entering your cancer program?
Our physicians in radiology set up a lung cancer screening process a couple of years ago, and we were getting small numbers of patients on a regular basis, really because of the reimbursement barrier. And in February, when the CMS developed a coverage determination, we took that radiology pathway with low-dose screening and we made it CMS compliant. We developed a connection with our clinicians in terms of an automatic order set and patient selection, so that a physician could order lung cancer screening, and it self-populated that the patient was an appropriate candidate. It made an automatic referral to smoking cessation, and that was sort of the front end load. And at the back end, we developed on the outside, we developed a CMS compliant screening registry, similar to what happens with breast cancer, using the Lung-RADS criteria and an automatic pathway for referrals, so that there was an appropriate handoff either to an automatic letter for a repeat screening if somebody had a small indeterminate nodule, and that went to the primary doctor, or a handoff to a pulmonologist if there was something that met criteria for diagnostic workup or biopsy. And I don't have the exact number, but it's already in the hundreds since we developed the pathway in February, tested it for a couple of months, and really made it available to our primary physicians broadly in June.
That's great to hear. Yeah, it would be great. I think I've heard that one of the projects and hopes is to sort of crowdsource and get information from all the different community hospitals on how they've developed their lung cancer screening pathways and processes, as well as how they've communicated that to the local community, primary care providers in an effective manner. So I'm sure we'll be hearing more about that. Another question that has come in is just regarding the futuristic sort of circulating tumor cells, the circulating cell-free DNA for molecular testing. How do you think that these tests are going to change the course of treatment or access for patients based on where these tests are today, as well as where they may be in the future?
I'm not an expert in this area, but I think the important thing about the cell-free DNA or liquid biopsies that we'll be able to utilize them for monitoring how patients do on treatment, so that you can't really biopsy a person repeatedly. We sometimes do repeat biopsies if we're interested in a new set of molecular markers in somebody who's progressed, but I think that this is going to make it more tolerable for patients to have this technology available.
Sure. And another question, we have time for this last one, I think, which is just general suggestions or guidance for community-based cancer programs in terms of how they can get more patients enrolled in clinical studies at their own institutions. Do you have any thoughts or suggestions or recommendations for them?
First, I think you need to have a culture that values clinical trials. The NCCN puts clinical trials as a level 1 evidence in many situations in people with advanced cancer, and I think that baseline education needs to happen for physicians and for patients. And then secondly, I think that people should take a look at their tumor registry, see what kind of patients they're seeing, and look for clinical trials that map to the biggest patient populations that they have. And then lastly, I think that I think that where we are in clinical trials, they should pretty much be molecularly driven, although that sometimes makes the patient pool very small.
Sure. Well, I want to thank you, Dr. Oyer, for joining us on this webinar, and thank you so much for your insights. And I want to remind those on the webinar, don't forget the resources that you'll find on the ACCC website, especially the resource titled Ongoing Advances and Improvements in Molecular Testing. And now I'll hand it back over to Daryl at ACCC to wrap things up.
Thank you, Dr. Kim. Thank you, Dr. Oyer, for joining us today. And thank you to all of you out there for taking some time out of your day to listen to this really insightful webinar.